Hello, I'm your host, Jackie Mullins, and welcome to From Cells to Us. How? The podcast where we figure out how life went from a single cell to complex creatures like us. So, wow, here we are, still in the Archean eon at about 3.5 to 3.8 billion years ago. But big things have happened, like huge, like something we'll always be talking about since it's still around. I want to do just a quick where we came from to where we are. In episode 1, we imagined we were a scared little RNA strand, if you remember. Shivering, scared little RNA strand floating in the primordial seas, watching organic molecule carnage all around us. Then we drifted into a fatty acid membrane, or a spherical Oreo. From there, we became more comfortable, more protected. We learned how to do our own chemistry, fold in on ourselves, grab amino acids, and have them work for us. Then, some of us lost in oxygen, paired up with some similar strands, and became DNA. Way more stable, a lot less mobile. Not to be outdone, we as RNA decided we're split in 3 ways. We'll be the messenger for DNA, we'll do the chemistry in the ribosome, and we'll bring over amino acids to be made into our workers, the proteins. This has been a long journey, a few billion years, in fact, but the transformation is nothing short of amazing. Now, here you are, the once scared RNA strand, all split up and running the cell with precision in LUCA. How could it get any better? Well, what if LUCA had kids? Last episode, we watched the genetic code get refined. Tina got her anti-codon ID badge. Rick got his protein crew and became a full ribosome. The wobble hypothesis gave evolution room to experiment. And LUCA emerged, the last universal common ancestor, the population of cells that would give rise to every living thing on Earth. But here's the thing about LUCA. LUCA didn't stay LUCA. LUCA had kids, and like all kids, they went their separate ways. They moved out. They got different jobs, developed different lifestyles, and eventually some stopped calling home. Today we're talking about the first great split in the tree of life. But before we dive in, let me give you a quick roadmap, because there are some new words coming and I don't want you to get lost. From LUCA to us, there have only been 2 major splits. That's it. 2 forks in the road that created all the diversity of life we see today. Split 1. LUCA's descendants branched into Bacteria and Archaea. That's what we're covering today. Split 2. About 1 billion or 2 years later, eukaryotes branch off from the Archaea line. That's us. For a future episode. So that gives us 3 types of cells. Bacteria, Archaea, and eukaryotes. I feel like these 3 are kind of like the Hemsworth brothers. A lot of people have heard of bacteria and eukaryotes, just like Chris and Liam, but not many have heard of Archaea, much like Luke, which is unfortunate because both are still fairly prominent. Luke, I'm sure you're listening. I thought you were great in Westworld. Now, here's one more word, prokaryote. Prokaryotes are simple cells. They don't have a nucleus. If you don't know what that is, don't worry, we'll get there. They don't have membrane-bound organelles, basically no little organs inside their cell. It's just the basics, DNA, ribosome, and a membrane. Both bacteria and archaea are prokaryotes. They're simple cells, the OGs, the ones that ran the planet for over 1 billion years before anything more complex showed up. Eukaryotes, that's us, we come later. We're the fancy cells with all the extras. Future episode. So prokaryotes are like a studio apartment. Everything's in that one room, your bed, your kitchen, your stuff. It works. It's efficient. There's no separation. Eukaryotes are like a mansion. Separate rooms for everything. Bedroom, kitchen, office, gym. Walls and doors are everywhere. More complex, more specialized, more space to do different things at once. The mansion comes later. Today we're talking about the studio apartments. Prokaryotes. The simple but wildly successful cells. But if bacteria and archaea are both prokaryotes, why do we call them different things? What makes them so different that scientists said, no, we need these in separate categories? Oh, and we need to talk about viruses because they've been here the whole time. Watching, waiting, infecting. And honestly, they might be older than all of this. Let's get into it. So LUCA had everything figured out, right? DNA for storage, RNA for messaging, ribosomes for building proteins, the genetic code, the central dogma, all the basics. But LUCA wasn't just one cell sitting there being perfect. LUCA was a population, probably a messy, diverse population of similar cells all doing roughly the same thing. And within that population, variations existed. Some cells did things slightly differently. Different membranes, different ways of handling energy, different strategies for survival, but one thing stayed the same: the universal language, the genetic code. That never changed. And that's going to matter a lot in a few minutes. But over time, these variations accumulated. The population started to diverge, and eventually, LUCA's descendants split into two major groups. We call them Bacteria and Archaea. Now, if you're thinking, wait, I've heard of bacteria, but what the heck is archaea? You're not alone, as we earlier discussed. We actually didn't even know archaea existed until 1977. A scientist named Carl Woese was comparing ribosomal RNA, the RNA inside ribosomes, our friend Rick, across different microbes. And he noticed something odd. Some of these so-called bacteria had rRNA sequences that were wildly different from actual bacteria, like not even close. It was like opening a book and expecting Spanish, but finding Mandarin. Woese argued that these weren't bacteria at all, that they were something entirely different, a third domain of life hiding in plain sight. People kind of thought he was nuts. It took 20 years for science to catch up to Carl Woese. Now it's in every textbook. Also, no one really talks too much about archaea because none of them cause disease in humans. We've just been living peacefully with each other. How about that? Unlike bacteria, where it's been an evolutionary arms race since day one. They evolve, we evolve. Antibiotics, resistance. On and on, the stakes just keep getting higher. Okay, so what actually makes bacteria and archaea different? Well, they're both prokaryotes. They both have the genetic code. They both use ribosomes. What's the big deal? Well, let's break it down. The biggest difference is their cell membrane. Remember that spherical Oreo in episode 1? The bouncer that was bad at his job? Both bacteria and archaea have membranes, but they build them completely different. It's like two people building a house. Same concept, walls, roof, keep the weather out, but one uses brick and the other one uses concrete. From the outside, it's similar, but under the hood, completely different. It's fatty acids versus isoprene chains, blah, blah. The point is different materials. But the archaeal membrane, it's tougher, more stable, handles heat, acid, and extreme conditions like a champ. That's why archaea dominate the extreme environments: boiling hot springs, super salty lakes, acidic volcanic vents. Many archaea are extremophiles. They can live almost anywhere due to their tough membranes. It'll be them, cockroaches, and Twinkies after the nuclear fallout. So the second thing that bacteria and archaea have different is cell walls. Rigid structures outside the membrane that give the cell shape and protection. And those too are made of completely different stuff. And perhaps you're like, wait, what, didn't we just talk about this? And close. But what we are talking about is the cell membrane. The cell wall is something different. The membrane is like the bouncer deciding who gets in. The cell wall is like the brick wall of the building itself. The bouncer works the door, but the wall keeps the whole club from collapsing. Bacteria and archaea have both, bouncer and brick wall. Your cells, eukaryotes, just have the bouncer, just the membrane. We're more flexible, but less armored. So bacterial cell walls contain peptoglycan, a mesh of sugar and amino acids. Think chainmail armor, flexible, interlocking links all woven together. Archaeal cell walls, no peptoglycan at all. They use different proteins and sugars, depending on the species. That's more like plate armor, solid pieces, completely different construction. Same job, different build. Bacteria's got chainmail, flexible, gets the job done. Archaea's got plate armor, heavier, tougher. Good luck getting through that. This is actually why antibiotics work on bacteria, but not archaea. Many antibiotics target peptidoglycan synthesis. No peptidoglycan, no target. And you might be wondering, why do we even know this if archaea doesn't make us sick? Well, back before we knew archaea were their own thing, scientists tested antibiotics on everything. When some, so-called bacteria didn't respond, that was actually a clue that something weird was going on. One more piece of evidence that archaea wasn't bacteria at all. And here's where it gets interesting. When you look at how bacteria and archaea handle DNA replication, transcription, translation, archaea actually look more like eukaryotes. That's us, the cells with the nucleus, than bacteria. Archaea's ribosomes were more similar to ours. Their DNA handling enzymes are more similar to ours. It's like bacteria went one direction, archaea started walking toward what would eventually become complex life. So archaea are not just weird bacteria. They're a completely separate branch of life, and they might actually be more closely related to us than bacteria are. It's like finding out Liam Hemsworth, the bacteria, was actually adopted, and Luke and Chris are the blood brothers. Even though Liam and Chris look more similar, the blood relation is between Luke and Chris, or archaea and eukaryotes. Wild, right? Okay, so why did LUCA's descendants split into bacteria and archaea? Why not just one type of prokaryote? Well, honestly, we're not entirely sure. But, just think about humans. We started in Africa, spread across the globe, encountered different environments, and adapted. Different climates, different food sources, different challenges. Given enough time, isolated populations diverge. Same idea here. LUCA's descendants spread out, encountered different conditions, found different ways to survive. Some went one direction, some went the other. Over hundreds of millions of years, those differences add up, and now they're two completely separate domains. And honestly, we're still trying to figure out the details. The split happened around 3.5 to 3.8 billion years ago, so we're doing forensic science on a crime scene older than most rocks on Earth. But what we do know is it happened, and both branches have been wildly successful ever since. So before we move on, let's talk about what life was like for those early prokaryotes, both bacteria and archaea. Like, what was their deal? Prokaryotes are tiny. We're talking 1 to 10 micrometers for most bacteria. For reference, a single strand of human hair is about 70 micrometers wide. You could line up 7 to 70 bacteria across the width of one single hair. That's crazy. And they're simple, no nucleus. The DNA just floats around the cell in a region called the nucleoid. No membrane-bound organelles, no mitochondria, no fancy internal structures, just membrane, cytoplasm, DNA, ribosomes, and that's about it. But don't let simple fool you. Simple does not mean ineffective. Prokaryotes have been running this planet for billions of years. They were here first, and they'll probably be here last. One thing that prokaryotes are really good at is making more of themselves. Some bacteria can divide every 20 minutes under the ideal conditions. That means one cell becomes 2 cells become 4 cells become 8. Do the math over a day and you've got astronomical numbers. This is why bacterial infections can get out of control so fast. And it's why prokaryotes can evolve so quickly. More generations mean more chances for beneficial mutations. Now, I know what you're thinking. Tons of kids without getting intimate? Sign me up. Well, slow down there, you anti-Casanova, you. There is a downside. They're all clones. When you reproduce asexually, you're just copying yourself. No mixing, no variation. Every daughter cell is genetically identical to the parent. That's great when times are good. You found a strategy that works, copy it a million times, take over, done. But when something bad comes along, a new threat, a change in the environment, a disease, everyone's vulnerable because everyone's the same. Did you ever hear of the Irish potato famine in the 1840s? Ireland was growing mostly one variety of potato, the lumper. Farmers kept planting clones of clones of clones, genetically identical. Then a pathogen called the potato blight showed up. And because every potato was the same, every potato was vulnerable. The blight spread like wildfire. Crops failed. 1 million people died. 1 million more fled the country. That's the danger of no genetic diversity. What kills one kills all. Prokaryotes face this same risk. If they only reproduce asexually, one bad virus or environmental change could wipe out the entire population. But at first it's great, right? You're multiplying every 20 minutes to an hour. If you're a successful bacterium, let's call you Brad, the bacterium. You've got good genes for surviving in your hot little thermal vent. You can make a whole colony of Brads. Brad Jr., Brad the Third, Brad the Revenge, Brad Forever. You get it. But here's where it gets dicey. The environment isn't stable. Earth 3.4 billion years ago was kind of a drama queen.
Whatever.
Temperatures fluctuate, chemical compositions change, new minerals appear, UV radiation is absolutely brutal because there's no ozone layer. The sun is literally trying to murder everything. And poor Brad and all his identical Brad clones, they're all equally screwed when the conditions change. But don't worry, they found a workaround. Keep this in the back of your mind. Here's where prokaryotes also shine. They can eat anything, and I mean anything. Some eat sugars, like us. Some eat sulfur. Some eat iron. Some eat methane. Some eat literal rocks. Some don't eat at all. They photosynthesize, capturing light energy like plants would eventually do. Prokaryotes figured out every possible way to extract energy from the environment. They colonized every niche on Earth. Hot, cold, acidic, alkaline, salty, deep underground, high in the atmosphere. Prokaryotes are there. This metabolic creativity is part of why life survived on Earth. Conditions were harsh and constantly changing. Having a diverse toolkit of ways to make energy meant something would survive no matter what. All right, so that was a lot of information. Let's do a quick checkpoint here, see what we got. All right, LUCA had kids, and those kids split into two groups, Bacteria and Archaea, the first great fork in the tree of life. Both are prokaryotes, studio apartments, simple cells, no nucleus, no fancy organelles, just DNA, ribosomes, and a membrane. But under the hood, they're completely different. Different membranes. They're brick versus concrete. Archaeas are tougher, which is why they dominate extreme environments. Different cell walls, chainmail versus plate armor. This is why antibiotics work on bacteria, but not archaea. Different machinery. And here's, the plot twist. Archaea's machinery looks more like ours than like bacteria's. Luke and Chris are blood brothers, Liam's adopted. We don't know exactly why they split. The same reasons humans spread across the globe and diverged. Different environments, different challenges, enough time. Evolution doesn't need a master plan. And both branches, well, they were wildly successful. They're still here, they're still thriving, billions of years later. Now, we need to talk about something that's been lurking in the background this whole time. Something that's not bacteria, not archaea, not even a cell. Something that might not even be alive. Viruses. So, when you hear virus, I'm sure most of you think of COVID, flu, that annoying thing you just have to let run its course because antibiotics don't work on it. But viruses aren't just a modern inconvenience. They are ancient, like ancient, possibly older than the bacteria-archaea split, possibly older than LUCA, possibly hanging around since the RNA world. And they've been messing with cells for billions of years. But why we haven't talked about them yet? Well, because viruses might not be alive, might not even technically be a cell. So what are they? Where do viruses fit in all of this? Are they prokaryotes? Are they eukaryotes? Well, they're neither. Viruses are weird. A virus is basically just genetic material, either DNA or RNA, wrapped in a protein coat. That's it. That's the whole thing. No membrane doing its own thing, no ribosomes, no RIC, no metabolisms, no way to reproduce on their own. If prokaryotes are studio apartments and eukaryotes are mansions, viruses are that stray dog that hangs around outside. He's not part of the house. He doesn't have his own place, but he keeps showing up, and he's wearing a sweater that grandma knitted for him. That's his protein coat. Mom thinks he's part of the family. He shows up every day. He responds to his name. He's basically ours. That's, some people think viruses are alive. Dad thinks he's just another mouth to feed, right? He doesn't pay rent. He can't survive on his own. He just takes our stuff and leaves. That's the viruses are not alive camp. The truth, viruses exist in this weird gray zone. They've got genetic material They can evolve, they can make copies of themselves, but they can't do any of that without breaking into your house and using your kitchen. They're not alive, but they're not alive. They're the freeloaders of biology, and they've been freeloading for billions of years. So in essence, viruses hijack your cell's machinery, your ribosomes, your energy, your nucleotides, and use it to make copies of themselves. Hundreds of copies, thousands, until your cell either dies, explodes, or gets so overwhelmed it can't function. Crazy to think that when you're infected, that's exactly what's happening inside of you. Silently, invisibly, your body is fighting off infiltrators of the highest magnitude, and you have no clue besides a runny nose. It blows my mind. Speaking of viruses and that blurry line between alive and not, remember Agent Smith in The Matrix? He calls humans a virus. Human beings are a disease, a cancer of this planet. You're a plague, and we are the cure. I'm bringing back imitations. Let's give it a whirl. But here's the delicious irony. Smith is the real virus. He can't exist on his own. He needs to hijack human bodies and minds to replicate. He spreads by infecting one host after another, turning them into copies of himself. No metabolism, no independent life, just endless copying until the system crashes. Sound familiar? Humans might multiply and mess things up, but at least we build, create, and adapt. Smith, pure parasite. Freeloaders supreme. So next time someone quotes that line at you, you can flip it. Actually, Agent, you're the virus. We're just the messy, wonderful hosts trying to wake up. So how old are viruses? We used to think viruses were relatively recent, like maybe they evolved after cells as escaped bits of genetic material. Like one day a piece of DNA just looked around and was like, I don't need you people, packed its handkerchief on a stick and walked out the door. No goodbye party, just vibes and a protein coat. Bye. But the more we study them, the more ancient they look. And that changes everything about where they fit in the story of life. And here's the evidence. Viruses infect everything. Bacteria, archaea, plants, animals, fungi. If it's alive, there's a virus that infects it. This kind of universal presence suggests that they've been around for a long time. Bacteriophages and archaeal viruses are very different. Bacteriophages are viruses that infect bacteria. There are also viruses that infect archaea. And these two groups of viruses are quite different from each other. Different structures, different strategies. This suggests viruses were already diversifying before the bacteria-archaea split, which means viruses might predate the split, which means viruses might be older than 3.5 billion years. Now, some viral proteins are incredibly ancient. Certain proteins found in viruses appear in viruses across all domains of life. These proteins are so old and so conserved that some scientists think that they may trace back to the RNA world. So when did viruses actually originate? Honestly, we don't know where viruses came from. There are three theories. One, they came first, hijacking RNA back in the RNA world. Two, they're escaped genes, that went rogue. Three, they're lazy cells that stripped themselves down until all that was left was genetic material and a coat. And the real answer is it's probably all of these. Different viruses, different origins. What we do know is they're ancient. They were here when bacteria and archaea split, and they've been shaping evolution ever since. Now, remember when I talked about the downside of asexual reproduction? How prokaryotes are all clones, and one bad virus or environmental change could wipe out everyone. I told you they found a workaround. I told you to keep it in the back of your mind, and here's the answer. Horizontal gene transfer. Instead of just passing genes from parent to child, vertical like a family tree, prokaryotes figured out a way to pass genes sideways. Neighbor to neighbor, stranger to stranger, completely unrelated cells swapping genetic material. That'd be like if we as humans had horizontal gene transfer and someone would be like, I love your hair. And instead of saying it's genetic and unattainable, you would say, thanks, I got it from a stranger I met yesterday on the sidewalk. Imagine how different our lives would be. And guess what was one of the main drivers of that? Viruses. When a virus infects a cell, it hijacks the machinery and makes copies of itself. But sometimes, accidentally, the virus packages some of the host cell's DNA into the new virus particles. Then, when the virus infects a new cell, it delivers not only its own genes, but the genes from that previous host. So it's basically like a molecular pollinator, a bee flying from flower to flower, accidentally carrying bits of pollen from one to the next. That's what viruses are doing, except with genes. Now, this is called transduction. It's one of the major ways genes move between bacteria, between archaea, and even between bacteria and archaea. And the only reason that could happen is because they have the same universal code. Viruses don't mean to help evolution, obviously. They're just trying to make copies of themselves. But in the process, they shuffle genes around. They spread innovation. They connect the branches of the tree of life in ways that wouldn't have happened otherwise. Weird bits of genetic material just trying to, stay not alive, who accidentally changed the course of evolution. And before I move on from viruses, I need to tell you this tidbit. About 8% of your DNA is viral. Not DNA that fights viruses, viral DNA. DNA that came from viruses. These viruses infected our ancestors millions of years ago, inserted their DNA into ours, and some of those genes turned out to be useful, so we kept them. One of these viral genes is essential for pregnancy. I am not making this up. A gene that originally came from a virus is necessary for the placenta to form properly. Without an ancient viral infection, mammals might not exist as we know them. So the next time you see a pregnant person, remember, that baby bump is brought to you in part by horizontal gene transfer. Locate an ancient virus near you. So you are 8% virus. Ancient viral sequences just sitting in your genome, mostly doing nothing. But, oh wow, if that sci-fi book doesn't just write itself. Okay, so let's get a little bit more into horizontal gene transfer, because horizontal gene transfer is actually the reason we're all here. Without it, life would have ended right here in the Archean eon. If prokaryotes had only used asexual reproduction, just making identical copies of themselves over and over, there'd be no variety, no backup plan. One bad day, one small environmental change, and that's it. Everybody dies. End of story. Roll credits. Now, when you think about inheritance, you probably think it's going in one direction, right? Parent to child. I got my grandma's eyes, my father's height, my mother's eczema. Thanks for that one, Ma. You get half your genes from your mom, half from your dad. They get half their genes from their parents, so on and so forth, back through time. This is vertical gene transfer, genes flowing down the family tree, normal stuff. But prokaryotes were like, no, we don't have time for that. I'm not buying anyone flowers. No one milkshake with 2 straws, thank you very much. They're stuck in the 50s, apparently. And with that declaration, they invented something entirely different. The horizontal gene transfer. This is completely unrelated cells swapping genetic material like they're trading snacks at lunch. Imagine you could walk up to a stranger on the street and just absorb their ability to play piano or speak French or digest lactose. That's basically what prokaryotes do, except instead of skills, it's genes. And instead of strangers on the street, it's other cells floating nearby. This is wild, and it changes everything about early evolution. So how does this actually happen? How do cells swap genes with random strangers? It's not like they're sitting around a microscopic poker table with microscopic visors on and microscopic chips in the middle dealing with their genes. I may have taken that joke a microscopic step too far, but I do like to picture cells playing poker, much like dogs. So there are 3 main methods of horizontal gene transfer. Method 1, transformation. This is the simplest. When a cell dies, it breaks open. Its DNA spills out into the environment and it just floats around. Naked DNA in the water or soil or whatever. And some cells can absorb that floating DNA. They just, slurp it up, incorporate it into their own genome. Oh, you don't need this gene anymore because you're dead? Cool, it's mine now. Now, can you imagine how different wills would be if humans worked this way, like actual legal documents, witnessed and notarized. And to my husband, I leave you my ability to remember every movie line I've ever heard. May you finally see what a gift it actually is. Oh, that would be fun. But alas, we do not work like that. However, a scientist found out how prokaryotes were working like that in 1928. This was actually how we first discovered horizontal gene transfer. Frederick Griffith was working with Streptococcus pneumoniae, the bacteria that causes pneumonia, and had two strains. One was smooth and deadly. One was rough and harmless.
Sup?
Griffith killed the deadly ones, just chopped them up. Griffith with a knife in the conservatory. But actually he used heat. The point is, these pneumonia-causing bacteria are dead. Now he took these dead pneumonia-causing bacteria and dumped them in with the living harmless ones. And lo and behold, the harmless ones became deadly. They stole the gene that made the deadly one deadly and incorporated it into their DNA. Griffith was like, excuse me, what? The scientific community was like, excuse me, what? That can't be right. But it was right. Bacteria were out here eating their dead friend's DNA and incorporating it into themselves, like some microscopic zombie apocalypse, but with genes instead of brains. Just insane. Now, method 2 is transduction. This is the one we discussed. Viruses acting like bees, pollinating the molecular world with mini gifts of new genes. You know, viruses. I'm on a roll this episode. Okay, and just a note, the viruses are usually bacteriophages, viruses that infect bacteria. And let me tell you, these things look crazy. They literally look like an alien spaceship. Google them later. They're terrifying and awesome. And just to use a previous analogy, a virus infects, Brad, the bacterium, uses Brad's cellular machinery to make copies of itself, and then bursts out like, a scene from Alien. But sometimes the virus accidentally packages some of Brad's DNA along with its own. Then it infects another bacterium, let's call her Brenda, and it injects Brad's DNA into Brenda. Now Brenda has some of Brad's genes. They've never met. They might not even be the same species, but thanks to a clumsy virus delivery service, Brenda is now part Brad. But wait, you might say to yourself, like, cool, the virus is gifting genes to others, but didn't you just say that the virus hijacks the cell's machinery and then leaves it to die? How will they be able to pass on these supposed gifts if they're dead? And yes, that is correct. That does happen most of the time. However, if that virus picked up some bacterial DNA by accident, it often can't complete a full infection. Why? Because that bacterial DNA took up some space where essential viral genes should be. Genes the virus needed to complete its hostile takeover now has bacterial DNA there instead. Now, instead of infecting and killing the cell, the virus just delivers some new DNA and leaves. It's like a Trojan horse, but, they forgot to put the soldiers inside. It's just an empty wooden horse, maybe with a gift. It's a little Monty Python and the Holy Grail energy right there. The third one is conjunction. This one is the most sci-fi-y. Two cells physically connect through a little bridge called a pilus. One cell extends this pilus, attaches to another cell, and transfers DNA directly. Scientists sometimes call this bacterial sex, but that's not really accurate. It's more like bacterial file sharing. One cell has a useful gene, builds a bridge to a neighbor, and copies the file over. No romance involved, no flowers, no milkshake with 2 straws, just pure utilitarian gene transfer. Okay, if you, like me, are like, what? With this one, I gotcha. The other two I get. I was like, okay, that makes sense. But then we have this portal just popping up and one cell's like, give me your genes. Like how and why? Well, some cells have these little extra circles of DNA called plasmids, separate from their main DNA, and some plasmids contain instructions that basically say, build a bridge, find a neighbor, copy me, send me over. The plasmid is the selfish one. It just wants to be spread. It has a separate drive from the cell. And the cell is just following the directions that the plasmid gives it. And sometimes that plasmid comes with gifts, like genes for antibiotic resistance or the ability to eat something new. So the receiving cell gets an upgrade it never asked for. The plasmid is also kind of like Han Solo in Star Wars, A New Hope. He's just in it for the money. I'm not here to save anyone. He's looking out for number one. But he does do some good. He saves a princess, what have you. The plasmid is the same. He's just looking to spread, looking out for number one, but sometimes accidentally delivers gifts. And here's the real world kicker. In 1959, when Japanese scientists figured out why antibiotic resistance was spreading so fast, it wasn't just evolution through mutation. Bacteria were actively sharing resistance genes. A bacterium in a hospital in Tokyo could share its antibiotic resistance with a completely different species, and that bacterium could share it with another, and suddenly you've got a superbug situation. Antibiotic resistance is now one of the biggest threats to modern medicine. The ancient past isn't so ancient after all. Now, quick PSA here. This is why it's so important to take all the antibiotics your doctor gives you, every single one. Here's what happens if you don't. Day 1, you take the antibiotic. It kills most of the bacteria. Day 3, you feel better. Day 4, you think, I'm fine. Why would I keep taking these? But most is not all. The bacteria still alive, they're the tough ones. They're the survivors. Maybe they had a gene that helped them hold on a little longer. Maybe they got a gift from a dying neighbor, a little resistance cheat code passed right over. Now those survivors multiply. And they pass that resistance to their kids. And suddenly you've got a whole population that knows how to survive that antibiotic. Next time you're sick, same medicine doesn't work. And here's the real nightmare. Those resistant bacteria can share that cheat code with completely different species through horizontal gene transfer. Your resistant bacteria meets a stranger, builds a little bridge, copies the file over. They're all swapping genes like it's a genetic flea market. Now it's not just one species that's resistant, it's spreading. That is how superbugs happen. This is why your doctors beg you to finish your antibiotics. So take the whole bottle, do not share with a friend or family member because you have the same thing. Kill them all, no survivors. Don't give them any help from us. Believe me, they don't need it. All right, so why should you care about horizontal gene transfer? Well, because it completely changed how evolution worked in the early world. Think about it. With only vertical gene transfer, a beneficial mutation has to arise in your lineage, then it spreads to your descendants, then their descendants, then slowly over generations it might spread through the population. But with horizontal gene transfer, if any cell, anywhere, figures out something useful, how to eat new food, how to survive a toxin, how to resist heat, that innovation can spread immediately to unrelated cells. It's the difference between one person inventing the wheel and teaching it to their kids versus inventing the wheel and immediately uploading the blueprints to the entire internet. Or like the Matrix, when Trinity needs to fly a helicopter and they just upload the skill directly into her brain. Done, instant pilot. I would absolutely love if humans could horizontal gene transfer like that. But alas, we're stuck with our slow, clumsy way of doing things. But for bacteria, evolution went from slow and steady to fast and chaotic. This is why the universal genetic code matters so much. Remember how I said it evolved once and got locked in? This is why that was so important. If every cell speaks the same genetic language, then any gene can work in any cell. A gene from a bacterium can function in an archaeon. A useful innovation in one branch of life can spread to another. The universal code made horizontal gene transfer possible, and horizontal gene transfer made early evolution fast. And just a quick jump into the future, our present, scientists are using horizontal gene transfer to our benefit right now. We may not get to download the ability to pilot a helicopter or know kung fu instantly, but genetic engineering, that's basically us doing horizontal gene transfer on purpose. We take genes from one organism and put them in another. Bacteria that produce human insulin for diabetics, horizontal gene transfer. Crops that resist pests, you guessed it, horizontal gene transfer. CRISPR, the gene editing tool that's revolutionizing medicine, we stole that from bacteria too. This is pretty cool stuff. So when you picture evolution, you probably picture a tree, one trunk, and that's LUCA. That branches into two limbs, Bacteria, Archaea. Then those branches split into more branches, no crossover, just up and out, right? Everything neat and tidy. But horizontal gene transfer means that branches are connected, genes jumping from one branch to another. The tree of life isn't really a tree. It's more like a tangled web, or, a tree where the branches occasionally fuse back together. This makes drawing evolutionary relationships really complicated for scientists. You can't just say this species descended from that species when they're both trading genes with random strangers for billions of years. It's like trying to draw a family tree for a family where everyone's adopted each other's children, but also kept theirs and remarried each other's spouses. Technically you could probably trace the lineages, but it's going to be messy. And honestly, the messiness is beautiful, right? Life isn't neat. Life is chaotic, creative, and constantly borrowing good ideas from wherever it can find them. So, here we are. LUCA has split into Bacteria and Archaea. They've spread across the planet. They've colonized every environment. They're swapping genes like they're in a genetic commune. Karl Marx may have studied bacteria and thought, well, if it works for them. Prokaryotes are evolving at breakneck speed. And this is the state of life on Earth for a long time. A really long time, like thousands of times longer than humans have existed. Prokaryotes had the planet to themselves for over 1 billion years, maybe closer to 2 billion. The first complex cells, eukaryotes, cells with nuclei, the cells that make us, plants, animals, they don't show up until around 2 billion years ago, maybe 1.5 billion, depending on which evidence you believe. That means for more than half of life's history on Earth, it was just bacteria and archaea, small, simple, everywhere. We tend to think of prokaryotes as primitive, as stepping stones on the way to real life, like plants and animals. But that's backwards. Prokaryotes aren't primitive. They're refined, optimized, incredibly successful at what they do. Your gut alone contains trillions of bacteria, on the same order of magnitude as the number of human cells in your body. There are more bacteria cells on Earth than there are stars in the observable universe. Prokaryotes aren't the opening act. They're kind of the main event. Everything else, including us, is just kind of a side project that happened later. All right, so let's do a checkpoint. Let's make sure we've got this. There was a split. LUCA's descendants diverged into two domains, bacteria and archaea. Same basic genetic code, completely different everything else. Different membranes, different cell walls, different ways of handling DNA. Prokaryote life. Small, simple, fast reproduction, metabolically diverse. No nucleus, no organelles, just the essentials, and it works. It works really well. Viruses, they're not cells. They're not really alive, just genetic material in a protein coat. The stray dog in grandma's sweater. They've been here the whole time, infecting, replicating, and accidentally shuffling genes between your cells. Freeloaders who accidentally shaped evolution. And 8% of your DNA is proof they were there. Horizontal gene transfer. Genes don't just flow from parent to child. They flow sideways, neighbor to neighbor, stranger to stranger, through dead cell DNA transformation, through viruses, transduction, and through direct cell-to-cell bridges, Han Solo, conjugation. Evolution on fast forward. Because horizontal gene transfer, beneficial innovations could spread across unrelated lineages. The universal genetic code made this possible. Evolution was fast and chaotic. The web of life. The tree of life is more of a tangled web. Branches are connected. Genes jump. It's messy, and that's okay. It works. So here's our conclusion. This is the age of prokaryotes. LUCA gave rise to two great lineages, bacteria and archaea. Different strategies, different structures, same fundamental code. And for over 1 billion years, they ruled. They spread across the planet. They colonized every environment imaginable. They swapped genes freely, evolving faster than anything that came before or since. This was the age of prokaryotes, simple cells doing extraordinary things. But something was brewing. Oxygen was starting to accumulate in the atmosphere, a deadly poison to most life at that time. Wild, right? Oxygen was poison. The world was about to change. Next time on From Cells to Us, cyanobacteria invent photosynthesis and accidentally poison the entire planet. The Great Oxygenation Event, the first mass extinction, and how life survived by learning to breathe the very thing that was killing it. Thank you for joining me on this biological journey. I'm Jackie Mullins, and this has been From Cells to Us. How? I'll see you next time.